To be very honest, I've learned so much about the failure in the lab. We like to say to PhD students, okay, look, even data which are not proving you are right are still providing information. One of the highest risk of looking data with some theory is that you introduce bias because you want to force data to go into direction you would like.
Hello and welcome to SciLux, the podcast where we talk about scientific developments and technological changes. Probably you're used to me saying in Luxembourg, but nowadays it's more Europe. So we often say that it's for the people who love science and those ones that don't yet know that they do. And we will again try to convince you today that it's really worth loving science with our guest, because our guest today is Professor Eleonora Macchia, who is Associate Professor in the Department of Pharmaceutical Sciences at the University of Bari and Head of Research at Åbo Academy University. She holds a Master's in Physics and a PhD in Chemical Sciences. So we'll be discussing both, I hope, and everything else you're doing. Eleonora, thank you so much for coming today.
Hello, it's really a great pleasure to be with you today.
And we are again recording not online, but next to the lab. So it's also a nice feeling to be there. Okay, starting from the very beginning, we're meeting also because you are one of the ERC ambassadors. And I wanted to start with that actually. What does it mean for you? Why did you become an ambassador?
Well, we are facing really a complex time where next generation are called to address very important challenges. And I really believe that the answer to this fundamental question is in frontier research, because investment in frontier research can, of course, be used in order to address those fundamental questions. So I really want to advocate for fundamental research and hopefully to convince policymakers in still investing money in those kind of frontier projects.
But that's very difficult, isn't it? I mean,
yes.
If I may, because, I do a lot of science communication, and it's always this question of, if you want to explain something, you need applications and you need analogies. And I remember when I was doing some quantum physics, it's like so difficult to have analogies because it's something that is so not based on intuition. Right?
You are right. But for instance, in my ERC project, we wanted to study how protein segregated into a film can communicate among each other. So it's really a fundamental kind of question. What topological transition those proteins can make under some environmental condition. But then we discovered that this amplification mechanism can be actually used to develop biosensor for in vitro diagnostics. So you see, we started with really frontier research, fundamental question, and then now we are at the stage of a clinical trial. So again, it's really important. There is no technological transfer without fundamental science.
That's a very good point that you're making. And you mentioned already one of the words, proteins. So I have to say, I always say it, I guess the listeners already know, I hated biology at school. I don't remember anything. And whenever I have to prepare for any kind of medical or biological subject, I get stressed because I know the words, but then I don't understand them. And one of it is that there is a lot of discussion, I guess also because of Google DeepMind, about proteins. Why do we care so much about proteins?
Well, actually, if you think about the development of an oncological disease, for instance, clinicians discover that even one mutated cell can determine the fate of our whole organism. And this thanks to the next generation sequencing technology that allow us actually to sequence most of the human genome. While proteome is largely unknown, it's like an iceberg, and we just know the tip, the more abundant protein. But nonetheless, our body produces a lot of mutated proteins, which are very important marker of particular disease. For instance, in pancreatic cancer, the diagnosis today is basically rely on biopsy procedure, and those are particularly invasive. And what new approaches are starting looking at the gene genome, but none of them is working on proteome. And we recently discovered that there is a particular protein which is called CD55, which is an extremely good fingerprint of high-grade precursor. So it's a good indicator that the patient needs to undergo surgery before this precursor becomes a cancer with a very low survival rate.
Okay, so you already mentioned the whole idea of screening for diseases and getting to know whether there is a chance of developing a disease right now. So first question is, how do we screen nowadays? So what do we check? Because you said we don't necessarily know so much about the protein. So what do we check?
Exactly. We are in an age where diagnosis still rely a lot on, let me say, art. So the competencies of the doctor, which basically screen the patient often when the symptoms occurs, which is too late. It's not preventive medicine, it's reactive sick care. So you have some symptoms, you go to the doctor and probably it's too late. And basically they look at your history and they start making more imaging exams. But typically in oncology, the limit of detection of imaging is already a tumor greater than 1 centimeter cube, which is already a huge tumor. There are, of course, laboratory techniques which are very much advanced when we think about genetic biomarker, meaning DNA, microRNA, and in this case we can rely on real-time PCR, for instance, or next-generation sequencing, so you can track those biomarkers down to the single molecule level, so the single copy of DNA, which is really effective. When we go to protein, well, we have ELISA, which is the workhorse in clinical diagnosis till the 70s, but it is completely blind when you go below picomolar, meaning that you need to have millions of protein in your droplet of whatever, blood, saliva, in order to get a signal which is reasonably higher than the noise. Recently, there are some development by Quanterix, but you track 1,000 of marker. So basically, it's not really in the preventive medicine regime.
And you think it can be?
We hope it can be. I mean, we are working to make it the case. This is basically one of the main goals of the ERC project.
To be able to basically get any type of sample that would be already enough to not give you noise, but actually some results. I'm still trying to imagine it, right? So let's say you get a droplet of blood, right? And then how do you get that single molecule of a protein, you said, that then gives you information that you need for, I don't know, future disease development or whatever else?
And now we go back to fundamental science then, because we basically rely on Mother Nature capability, because our sensor has a detecting interface where you put 1 trillion of capturing antibody there, which are capable to interact spontaneously with the biomarker you want to analyze. It can be an antigen, but it can be even, if you use a single-strand DNA, the complementary strand to the one you want to detect. So basically, when the interaction between one biomarker and the ligand, so the protein segregated on your detecting interface occurs, then this protein is for some reason capable to share the news to the neighbor. So it's like the protein, they have their own language, so they can communicate among each other, and there is this very tiny interaction, one-to-one, so one marker, one ligand, but the news is immediately spread, like in a bus in the rush hour. When someone enters, everybody feels it, even in the back of the bus. So it's more or less the same, and you can get a change in the electronic current, which is higher than the noise. So we basically rely on Mother Nature competencies.
Before we continue, I was just thinking right now, when I introduced you, with your master's degree and your PhD, okay, chemistry still, but it was started with physics. How did you end up in medicine? Was it because, as you said, you started fundamental science and then it actually led you to discussing proteins and DNA, or?
I did my bachelor degree actually in Bari, and I was at the moment where I needed to choose a topic for my bachelor thesis. I was very much fascinated in electronic, but I didn't want to study standard inorganic material, silicon, so I decided to ask my thesis in the Neighbor building. So I went to chemistry, and Professor Torsi, she's a pioneer actually in bioelectronics. So I went there and I said, look, I have searched a lot. I saw that there is a lot of modeling on MOSFET technology. Can I join your group? And started studying modeling of organic material, so organic transistor. And she was thrilled. I joined her group, and actually we started a collaboration that still lasts nowadays. And it's very funny because I started with her integrating biomolecule into the transistor, looking at the modeling, so like really a physicist. Then I spent my PhD in the lab, and then I'm back to the model. I spent my postdoc in Finland. I started chemometrics, which is in a way the old-fashioned way of calling artificial intelligence. So it's basically statistics applied to chemical data. And I came back home to Bari at the end of this journey, and I joined back my original group, bringing those competencies. So it's a very, funny story in a way.
Absolutely. But it's also interesting because we tend to, at school separate these so strictly, right? If you do biology, you do biology. If you do physics, you do physics. And then it turns out that you can jump actually pretty easily, or it's just you?
You can jump. The system is now encouraging younger students more and more, to mix and match competencies. Yes. But if you think about the concept of circular Earth, it is there till the 80s, actually, till the previous century, actually. We discovered it, of course, during COVID, because the pandemic made us realize that we cannot think about health like human health, and that's it. We need to take care of our whole system, because at the end of the day, we are a closed system, nothing more than this. So, of course, this pushes us to put together physics, chemistry, medicine, vet, even architecture, law. A circular Earth means that we need to sit around the same table and start facing the new challenge with a multidisciplinary approach.
And that also leads me to convince our listeners that if you're interested in this discussion, you can also check out all the other episodes where you can learn about other sciences and then maybe jump ship, as you're saying, right? Because it's closer than you think. It's just you have to start somewhere, just like I'm sure you did at a certain moment when you had to figure out the DNA and proteins and whatever else, right? You mentioned that the chemometrics, right, was kind of like a small sister or precursor of artificial intelligence in a way, right, because it's statistics and all that. So I think it's a good moment for you to ask the pub quiz question that you prepared for our listeners. And listeners, remember the answer only at the end of the podcast.
The question would be, what is the role of artificial intelligence into predictive medicine?
And it's not because you studied it before a bit, but something else, right? So we will be discussing it later. I also wonder, is it a bit related to what I touched upon, so the protein folding, or not?
It is.
Okay, good. So I'm not going to ask that right now. We can discuss it further later on. But I am very confused right now. One of the reasons I'm confused is the fact that you mentioned the word transistor. Mm-hmm. And for me, transistor is, IT, computers and whatever. And now suddenly you're talking about biosensors. Like, what?
You are right. Basically, our device, we develop actually in two kind of format. One, it's a single device, which is basically enough if you want to make a qualitative diagnosis of an infectious diseases. So you want to track a single virus or pathogen. For instance, we were working a lot with Xylella fastidiosa, which is a main issue in the southern part of Europe, especially in Italy, but it's not enough if you want to work with with pancreatic cancer detection, because in that case you cannot look just at one single biomarker, but you need to develop a panel of different markers you want to analyze. So, but in any case, you basically have what is a biosensor? It is a tool comprising a detecting interface, which is the one that takes care of actually hosting the binding events. So you need the biomarker to interact with your device. Once this biochemical interaction has occurred, you need to convert it into a signal you can actually measure. And this is the role of the transistor. So you basically couple a cartridge which hosts the detecting interface into an electrode, basically. And this is coupled with the reader. The reader is basically made of transistor, so they read a current level before anything occurs. Then you put your droplet of saliva or plasma or serum, whatever. These biochemical interaction occur, and all of a sudden you see a change in the current level registered by the transistor. And this part is like a mobile phone. It's like a reader.
And does it mean it can be done at the patient's house then? Because I noticed that you were talking about this whole idea of point of care testing, right? So you don't have to go to the hospital, but you could have a device at home.
Yes, at this stage I see more feasible in a doctor cabinet, for instance, or in a drugstore. But actually, yes.
Ideally,
yes.
Okay, but then it's very different than the tests we can buy in the pharmacy, right? Because going back to, again, what we already have, you can talk about, I don't know, pregnancy tests, right? One of the things that I guess most of the women are aware of. Then you have now, because of COVID, we all know about the COVID tests, but that's different, right? There are no transistors in there or whatever. It's just you put certain type of substance, I don't know, it can be urine or blood or whatever, and then it's just a kind of a biochemical reaction that you get.
And you get an optical signal in that case. But the main issue with lateral flow tests nowadays...
That's how they are called.
Yes. Okay. It's the reliability of the test, because basically it has a lot to do with statistics. Again, we are always playing with what we accept as a significant signal, right? Because first of all those sensors you were mentioning, they basically have a limit of detection, meaning the threshold, which defines a device, the yes or no output of the assay. This threshold is quite high. It's in the micromolar range. So you need to get, if we think about COVID, an infection which is already in an advanced stage in order to tackle it. But if you think about your experience during COVID, of course, the main issue is related to the false negative rate, because if you were tested positive, then the chances of being a false positive were below 1%. But if you were a false negative, you could reach up to 50% chances of being actually a false negative. So it's like, playing with a coin.
Yeah, totally. But why is it like that? It's just that the concentration in the blood is not enough.
No, it's actually related to how you keep your threshold statistically, because what is the limit of detection is typically defined as you take a lot of blank experiments, so there is no biomarker in your biofluid, and you collect a signal which has a Gaussian distribution. Now, you define your threshold moving 3 sigma away, 3 standard deviation away from the average of the blank. With our device, and these by designs give you a 50% overlap of the Gaussian distribution of the load with the Gaussian distribution of the blank. But if you move this threshold, as we are doing with the SiMoT technology, 6 sigma away from it, then you get less than 1% chances of getting both false positive and false negative. So it has a lot to do on how you treat your signal.
Okay.
So again, chemometric.
Again, going back, right?
Going back.
Important. Okay, so let me now, as you mentioned and talked about it a little bit more, talk about SiMoT. As you said, SiMoT means what?
Single molecule with a large transistor, but in a Vietnam language, it is like eating the rice till the very last rice. Yes.
Okay. How
till
did you?
The very last. Yes.
So it's on purpose.
We discovered it actually afterwards. We first give it the name from single molecule with a large transistor, so it was nice to hear. But then we discovered this meaning.
Okay, so single molecule, I already understand, you explained, but why large transistor, not small?
Yes, because basically here you are opening a very fundamental research question again, because the idea till 10 years back was the following. If you want to track a single molecule, you need to work with the detecting interface hosting few bioreceptors. So this approach were actually called near field sensing, and basically what they do, they capture a train of single binding events. But although these approaches are very interesting from a fundamental point of view, because still you get a signal from single interaction, you are very much limited by the diffusion barrier. So in order to get a reasonable chance of your biomarker to actually find the 1, 2, 3 biomarker on the detecting interface, you need to work with a very high concentration of marker into your solution. Then you are not limited anymore by diffusion barrier, and you tackle this train of single binding event. So you have a single molecule resolution, not single molecule limit of the detection. Of course, when you work with the patient fluid, you cannot choose the concentration of biomarker. It is what it is. And if you want to make preventive medicine, actually, you are willing to work with very diluted solutions. Even in cancer, for instance, a tumor with a volume of 1 centimeters cube basically produces, put into the bloodstream, around 10 to the power of 3 copy of DNA per ml, and of those, just 1 is probably mutated, which is the one you are tracking. So you need really to have not single molecule resolution, but single molecule sensitivity. But now the point is, how can we avoid the diffusion barrier? So we increase our detecting interface. We invert basically the ratio between the marker and the capturing antibody. You have few marker on your solution, so you need to get trillion of capturing antibody on your detecting interface. That's why large.
Okay, I understand, because of course for me large means the size, but transistors nowadays are small, right?
Yes.
Anyway, so it doesn't mean it's like, the old computers that take the whole room.
Actually, the cartridge is not that small. It's the electrode has a detecting interface. I mean, the diameter is 5 millimeter. So there are 10 to the power of 12 capturing antibodies over there, packed really close, like Mother Nature does on the surface of a cell.
I know as a researcher, you don't necessarily like to talk about challenges because, it's also your funding and whatever you are planning to find out. But what's the main challenge for SiMoT?
Well, now we are facing clinical trial. So the first challenge, we are challenging on purpose the system by analyzing 1,500 biofluids. They are actually plasma, serum, and urine collected from oncological patients. And we are adding into those fluid exogenous marker, very diluted at the single molecule level, in order to check whether our model withstand this increase in the dimension of the data set. We are also developing onboard quality control. So we want also to add into our data analysis pipeline process monitoring, because of course those are complex systems. Something might go wrong during the analysis, and we want to make a warning to the clinician telling, okay, okay, look, this test is probably invalid. It's better if you repeat it because statistically this is an outlier, possibly providing feedback on what went wrong during the analysis. And of course, the big research question is why we are so sensitive. So we are trying to deep into the amplification mechanism with other complementary technique.
Isn't the challenge also the biomarker itself? Because in your case, you mentioned pancreatic cancer. Are we really sure that that protein is the one that is the biomarker for that cancer?
You are right. We are actually a targeted approach. So we need to know which biomarker we want to look at. There is another very active research field in finding the most representative biomarker, and those untargeted approach mostly rely on mass spec. So it's a tough job.
Oh, yes, of course. I mean, I'm not trying to put you, in an uncomfortable position, but I guess any research is a tough job, right? And it's, as you're saying, it's also about the failure part, if you want, right? Testing and actually not trying to prove that you were right from the start, because that's not what research is about.
I agree. And besides, I don't look it as a failure, but it's a learning process. To be very honest, I've learned so much about the failure in the lab. So we like to say to PhD students, okay, look, even data which are not proving you are right are still providing information. And one of the highest risk of looking data with some theory is that you introduce bias because you want to force data to go into direction you would like. So as a chemometrician, I always try to be unbiased to say, okay, let's try. I'm not expecting anything. This is something when PhD students ask why we are doing this experiment, what we do expect, we don't expect nothing. We run the experiment, we look at the data, and without any bias, we try to analyze them and see what those data are telling us. So it's not failure, it's science.
Well, this is a very good approach also because, before we started the discussion, we both said we have kids. And, when you go back to school, I guess school rather gives you the expectation is that you have to answer according to the expectation, if you want, right? So the answer is already prepared. And I notice also when I go to schools that sometimes students are pretty confused when I ask them questions where I don't know the answer of, right? I think maybe we should start a bit earlier to prepare the generations to actually be ready to have questions that they don't know the answer of and be ready to be asked these questions.
Probably this is one of the reasons why, on average, less students choose a STEM career path. And this is a huge problem because actually STEM careers are becoming more and more important for our society because in every field, even law needs to be supported by data. So we are in the area where artificial intelligence is pretty everywhere, and we have a problem of alphabetization of the population on this topic. So I think we shall probably change from primary care, from primary school, the way we train students. We as adults must put a lot of effort in supporting their critical mindset.
And as we went a little bit outside, let's say, had a meta-analysis, let's do a meta a bit more. Because I have a chance to talk to researchers in so many different countries, I often like to ask them, so how is it to do science? And you have an amazing opportunity to compare. I don't know, should you even compare south of Italy and Finland? But maybe yes. So how is it to do science here in Bari and how is it in Finland?
Well, I'm usually an optimistic person, meaning I like to see positive aspect wherever I am. So Finland was really a great experience because I had the chance to explore, really to delve in an European environment because I had an amazing research office really supporting into the writing of EU project. So this was really, really important. And besides, besides, I have the chance to explore a different environment and find my own career path. Because, of course, after PhD, you are not totally independent. You need to find your own competencies and something that is really your own career path, different from what you have done before. So Finland helped me a lot in finding my own way, I must say. Coming back to Italy, well, I did it mostly because I'm the result of public investment, because all my research career was done in a public school. So I cost a lot of public money to support me until a PhD. So I decided, okay, now I did my experience abroad. I've learned a lot. I was exposed to a really European advanced environment. Now it's time to come back and give those money back to my country some way. And it was also important in order to learn how to be a PI, really, because what I have found in Finland is that they have a very good balance between private and work life. And I have also learned how to make the work environment really pleasant, like a second home, like with the kitchen infrastructure, for instance,
Okay, so what you're saying is that this is not typical here?
No, it's not typical. And what I also appreciated is that it's not really work-related, but the way Finnish people handle the childcare, because they have a paternity leave and maternity leave, which are perfectly balanced between mom and dad. So usually mom take the first 5-6 months, and then the father goes on child leave, and this really is the game changer when we think about filling the gender gap.
I totally agree. A couple of months ago, I was interviewing a researcher in Denmark, Søren, and he was there also during the conference. This conference it is called EURIPS, and it's a kind of a sister conference of NeurIPS, which is an American machine learning important conference. And they decided to try to have kind of an European alternative. And there was a big discussion on why we should do it, whether it makes sense and whatever. And he was on stage with thousands of people listening to him, and he said, one of the main reasons I don't want to go to the US anymore is because I have kids. And I was like, my God, a male researcher saying this,
Yes.
I mean, this was the first male researcher I heard, because many of them say, oh, it's so great, I can travel so much. And it's true. When you research, you have the opportunity, right? Amazing opportunity to go to conferences, whatever. But when you have children, maybe you don't have that opportunity anymore. And somehow it does sometimes land on women more than men.
Exactly. This is most of the case. Women colleagues, they are most affected. And our academia still rely a lot on networking, because you need to be in conference to meet people, enter consortium for EU project, especially after PhD, when typically kids comes by. And this is affecting mostly women. So really, I strongly believe that the only way to solve this issue is to share perfectly 50-50 the balance, the childcare. There is no other way.
Yes,
to offset.
That's going to take quite some time, I think, but let's hope for it. And going back to, networking, as you mentioned, so just still the research conferences, because I try to go to quite a few. Sometimes I understand something, sometimes I have no idea what is discussed, but I'm trying very hard. So what kind of conferences do you go to if you get to? As a PI, I guess a bit less nowadays, right? Or you still go?
Well, no, I'm still joining, and especially after the ERC, we do have, of course, dissemination obligation. So it's I really like to join conferences, especially broader one, when you have the chance to follow symposia that are not really related to your own research, because they can open a bit your mind. Sometimes we are stuck on our topic.
Yes.
We are doing chemometrics, we do to chemometric conference, sensor conference. But if we open up a bit our mind, sometimes new idea come by. And I really like to join broad conferences.
So what was the last idea that inspired you then?
We moved recently from electronic kind of biosensor to optical one, because we were really exposed to the surface plasmon resonance community. The idea was, look, we have this amplification mechanism that has nothing to do with the kind of transducer we used, but we discovered there is a nice paper. It's a review paper we published with Professor Torsi and I on a Nature review. And we basically discovered that there are a lot of paper out there when they reach these signal to noise ratio. So basically they are capable to track few molecules on a large surface. And what this different protocol have in common is that the biolayer film, so the receptor film, undergoes a pH shift. So we basically had the idea that this pH shift might trigger the amplification mechanism, and we decided to apply to an optical technique, and it actually worked.
So it's good to go out of your comfort zone, as you're saying, right?
Yes,
exactly.
yes.
In this particular case.
And besides, it's boring to be always in your comfort zone.
No, of course. I mean, at the same time, it's also in that small area deep in, you can turn a bit left, but I prefer personally to be a bit up and turn left more radically in this case.
Yes, I think it's difficult because this means that you really need to push yourself to go to the basics. I mean, you need to look at the essence of the phenomenon you are looking at without overstructure, which is not necessary. Simplifying is not necessarily the simplest thing to do.
Absolutely. And also, I remember, again, I don't know why I keep coming back to this Europe's conference, but there was one keynote of a professor that has always studied the same methods and for machine learning. So, it's this field that has a lot of hype right now. We will come back to it in a second for the pub quiz question. And he kept talking about something where he was a pioneer in 30 years ago. Right? So he's still that method that he came up with 30 years ago. He is kind of processing and trying to reapply and convince people that actually that method can be still good, whereas everybody is going the other direction. And he might be right. I'm not saying wrong, but it's a bit sad, right? That you kind of keep going back in circles where you could be also applying your knowledge and abilities to another field, let's say. So as I already hinted at it, I think it's the moment to wrap up our discussion and solve the pub quiz question. So we are going back to artificial intelligence, right? So if you can remind our listeners what the question was about, and then let's solve it.
So how artificial intelligence can support a clinician for preventive medicine? And well, we were talking about how that diagnosis is actually done nowadays, and we were talking about heart. So the clinician putting together a lot of information. Now, the idea is to provide clinician with some data-driven diagnosis. And besides, the idea is that the clinician can actually rely not just on the outcome of your single analysis, but he can compare your analysis with millions of other analyses. Of course, this doesn't mean that we can substitute the doctor with ChatGPT. This is an important aspect because the mantra in chemometric is basically whatever model you train is garbage in, garbage out. So you need to, why we still need science scientists to train those models and supervise those models, because you need to feed the model with really good information. With good information, I mean with a physical model. So you need to really master the technology and the physics behind it.
Yes, absolutely. And this is, of course, a science podcast, so we do talk a lot about data and data collection. And there was a very interesting discussion with Carlos Vega about it a couple of months ago, where we were talking exactly about the problem of models that were supposed to predict, I don't even remember what it was, one of the diseases, and they were trained on pictures that were collected somewhere, mislabeled, and then suddenly there's this whole hype. I think one of it was COVID. So it was some kind of chest pictures of something, and then it was supposed to give you, positive, negative results. But exactly, the whole model was based on very bad data. And that's right. Kind of our age right now. The data, we start with the data, not the model.
Yes, and also I'm also afraid about a bit the data scientist. I mean, the risk...
It's good I'm not one, because I was planning to become... So we'd be afraid of it.
No, I mean, it's okay, but it is difficult to analyze data that you don't understand, because the risk is that you train the model and you don't really know what the model is doing. If it's taking garbage from this picture, or if it is really looking at features that have physical meaning. So this is why it was really important for me to spend 3 years fully in the lab, because I get the chance to know really well the physics behind the transistor. So now when we train the model, we know exactly how to feed it in order to get significant feature, right? So you change the data set and the model is still working, is robust enough, and you are not bumping into overfitting, let's say, issues.
Yeah, of course. But then the data in clinical trials is not your data.
We are actually the sponsor of the clinical trial. We were founded by Regione Puglia, so there was a public effort in supporting us, and the clinical trial are actually run with our technology at the oncological hospital in Bari. So I'm lucky enough, I'm very proud of being the biostatistician of the study. So I'm really looking forward.
So what's, just to wrap up, what's after the clinical trial? What are you hoping for? Or I shouldn't ask, it's too early to answer this.
Well, we hope to get the European validation of this technology, and we hope to see it in public health hospitals, so that everybody can actually benefit from this technology.
Of course, so fingers crossed, and hopefully in a couple of years we can discuss another technology that you'll be working on, because that one will be already done and dusted and in all the hospitals.
Hopefully.
I'm a bit positive, maybe too much, but I'm keeping my fingers crossed. Thank you so much, Eleonora, today.
Thank you.
And this is it for today. Don't forget to subscribe, to follow us. We are basically on all the possible social media and podcasting platforms. My name is Hanna Siemaszko, and this was SciLux.